Really got many thanks for the kind introduction and for the invitation to Berlin. I would like to give a bit of an insight into the mechanism also of non-viral vectors, but also what to consider when you deliver different types of nucleic acids. So it's a bit about the selection of the right transfection reagent, about the mechanism, and also its in vitro and in vivo application, but do not expect transfection protocols. That would definitely take too long, and every company that sells a transfection reagent always states that it's the best, of course. So the advantage of such a synthetic delivery system is that you can really upscale it. It's a more or less simple chemical. You can synthesize it, you can produce it, ideally in the gram scale or more. You can add extra features, so kind of responsiveness. So that means, for example, that at a certain time during the transfection process inside the cell, the payload is released. You can attach cell-binding ligands. There is pretty much no limitation for the size of the nucleic acid to be delivered, but still, it's still a bit hampered by its efficiency. Yeah? So viruses had time to evolve for millions or billions of years, and these are more recent developments, although they are already on the market now for a few decades. Still, one has also to consider the cellular toxicity of such components, because you add a lot of material onto your cells, so you inject it into an organism. And another bottleneck is definitely the in vivo delivery. So how can you choose? There's a broad field of commercial delivery vectors. It really depends on the cell type and on the nucleic acid type that you will deliver. Well, I will then come to the mechanism of nucleic acid complexation and also the cellular interaction, the release dynamics, because it is a dynamic system. Then a bit about polycation-based systems and receptor targeting, and then I will switch to in vivo applications. So how can you measure relatively fast in vivo kinetics of your non-viral vectors, and which type of imaging method can you apply? In the field of cationic lipids, you have a very broad area to choose. A very broad range to choose, but the principle is always the same. You have a cationic head group, you have a linker molecule, you have a hydrophobic part, and as you can already see here in the head group, for example, of lipofectamine, one of the first versions, you have nitrogen groups. Here you have amines, primary, secondary, sometimes also tertiary amines, and they are also important for the processing inside the cell. On the other hand, you have polycations. So they can be relatively simple, like polylysine. You see it here, this epsilon amino group. It's always positively charged with a pKa of about 14. Or you can have a mixture of primary, secondary, tertiary amines, like in the case of branched polyesylenimine, or also more defined structures are available, dendritic structures, so they are per definition not polymers, but they have a unique molecular weight. So how do they interact with the nucleic acid? There has been some work done already, it's been calculated 20 years ago by Joachim Redler, and he showed very nicely that such cationic lipids and nucleic acids form a kind of sandwich, kind of lamellar structure, and then depending on the conditions, this can also be a hexagonal structure, like here this inverted hexagonal structure, or this hexagonal structure, which forms a kind of certain pattern. Yeah? In the case of polycations, it's not that precisely defined. There was some, or there is some work done about the molecular modeling, about the interaction kinetics between polycations and the nucleic acid, and there is one proposed mechanism, so that initially you have a have this ionic interaction, and then a hydrogen bonding occurs between the polyethylene imine and the phosphate groups in the backbone of the nucleic acid. So this is how these particles form, and it happens pretty much spontaneously, and you can control it also. Salt concentration, for example, that you have in your mixing buffer, or with the molar ratio between the polycation and the DNA. Then you have your particles. So how do they enter the cells? First, most easiest way is if you have a surplus of positive charge, you will have electrostatic interaction with a negatively charged cell surface. And there, mainly heparan sulfate protein glycans are involved. You can also attach ligands. I will come to this later for receptor-mediated endocytosis. You can enhance this binding. In cell culture, it's relatively easy. You can centrifuge your plate. You can also ensure that you have no competitors in binding to these heparan sulfate protein glycans. For example, in excess of a polycation or excess of cationic lipid, or serum proteins which will pretty much destroy your transfection complex before it enters the cells or works as a competitive inhibitor. There was one process proposed by the Jean-Paul Baer group already quite some years ago, and was also confirmed by other groups. So after this electrostatic binding, you have a relatively dynamic structure here. So actin, and actin is ordinamine, and both are bound via such kind of attachment proteins that bind to the plasma membrane from the inside and then drag this particle into the cell. Yeah? And this then forms a new vesicle inside the cell. So this is if you have binding via HSPGs. The other option is you add a specific ligand. So what many labs did, and we also do, is we form molecular conjugates. So this is relatively simple chemistry. You have a binding moiety, should be a polycationic charge on it. Then you can use a linker molecule, which at the same time can shield the excess of positive charge, first of all to increase your specificity, but also then later on in vivo to prevent binding of non-target tissues and blood components. So this can be polyethylene glycol, and on the distal side you can bind your ligand. Can be a peptide, can be a small protein, can even be an antibody, but please ensure that the ligand is not too big, because this can then also negatively influence your particle formation process, and then you're pretty much free to use any kind of nucleic acid to condense it into virus-sized particles. I will bring one example as a maybe potential pitfall or something that you should take care of. The epidermal growth factor receptor. Yeah? So EGFR, you find it upregulated on a broad range of tumors, so it would be very good target for tumor selective delivery of your transgene. But when its ligand EGF binds to EGFR, you will have activation of this signaling pathway. So you have downstream signaling, you have phosphorylation of ACT and ERK, and this at the same time induces macropinocytosis, so you have very fast and efficient uptake of your particles, but this will also cause a mitogenic effect. And in case of cancer, might be not really this what you want to feed the tumor with a growth factor. So what we and others did, we designed and then synthesized EGF receptor binding peptides that ideally do not interact like EGF with EGFR, but bind on at another part of the receptor. And so we managed it, or could demonstrate, that such particles, here this is with this peptide, and this is such a gene transfer particle here with EGF as a ligand. Here with EGF you have activation, so you have phosphorylation of ACT. Here you do not have phosphorylation of ACT or ERG. So you have internalization, and this is due to the constant internalization and kind of recycling of the EGF receptor, so it's a bit slower, but you do not get activation of this pathway, and in vitro but also in vivo this gives you a positive effect on the transfection efficiency, so it selectively increases your transfection efficiency. So okay, this is the uptake. Endosomal release is another really big bottleneck, so it's important both of course for DNA and for RNA. In case of if you deliver messenger RNA or siRNA, it also should be released already there so that it's accessible in the cytoplasm. And there are some proposed mechanisms. So for polycations, but maybe also other components which contain protonable amine groups, the so-called proton sponge mechanism is proposed. So you have these ATP-driven proton pumps that lead to influx of protons into the endosome and acidification. At this, then your polycations will buffer it. Yeah? They will pretty much soak up the protons, the secondary and tertiary amines. You will get a polymer swelling. Then you will have promoted chloride and further proton influx, and then an osmotic effect. So your endosome will somehow swell, and at a certain time point you will obtain endosomal rupture, more or less efficient. And this can also be measured. This, like here, old work from the group of Frank Zucker. You see this increase in endosomal volume due to this buffering effect. Slightly different is the mechanism with cationic lipids. Here you usually also have internalization into intracellular vesicles, but then intermixing process, a kind of flip-flop mechanism takes place between the negatively charged lipids in the endosomal membrane and the positively charged cationic lipids in your lipoplex, and this then in the end leads to release of your payload and rupture of the vesicle. But what happens with the DNA or with the siRNA if it's in the endosome? Is it still intact? Is it alone? Is it freed up or still condensed? And there was a quite interesting experiment colleagues of mine did in Munich. So cells were co-incubated with a photosensitizer and then also with gene transfer particles, in this case plasmid DNA and linear polyacylenamine, both labeled with different dyes. And then they took a laser and activated this photoinducible structure inside the cell, and that then immediately these particles burst, or these vesicles burst. And after this induced vesicle burst, you can see that here, and this is the DNA, you see a kind of cloud of plasmid protruding into the cytoplasm. It's relatively slow, so it looks like that it's non-degraded. And independent of it, you also have release of the polyacylenamine. And later on, another group, it was shown that both such polyplexes but also lipoplexes do release their payload relatively fast, even without such a laser induction. So this is also important. It has to be a dynamic system. It has to be released, so the cell has access to the free nucleic acid. Another thing which can be a pitfall will be now presented as a kind of example. Polyacylenamine, I already mentioned it. If you form these particles with it, inject it intravenously in a mouse, you will get a lot of transgene expression in the lung. It's mainly the type 2 pneumocytes and endothelial cells. It works with luciferases transgene, with beta-gal other stuff. But please note, these proteins, they are all cytoplasmic, yeah? They just swim around in the cytoplasm. And there was a quite interesting effect we observed when we used, so this was the group of Christine Spitzweg and my research group. We used the sodium iodide symporter as a transgene, as a therapeutic gene, also as a theranostic gene, because this is a transmembrane protein with several transmembrane domains, and it transports iodine into the cell. Yeah? So if you look for iodine transport in a mouse, if you inject radioactive iodine and do a gamma camera imaging, you see here, so this is the nose of the mouse, so this is the tail. You see a lot in the thyroid, you see something in the stomach and also in the bladder due to renal excretion. So what we did next is we took these polyplexes that I've already shown you, which cause a very strong transgene expression in the lung based on luciferase, and in the gamma camera we saw nothing. Yeah, so we were quite surprised, cannot be, this was our positive control, but we then also checked the mRNA levels for NIS, and there we found a lot of mRNA in the lung. And this is maybe also a good take-home message. Think about it, where is your protein that they're going to express? Will it be just somewhere in the cytoplasm? Does it have to be secreted? Is it in the membrane? And we concluded from this data that this polyethylene imine also induced the membrane damage and somehow didn't allow a proper incorporation of this NIS protein into the membrane. So that brings me already to the in vivo application. And here, a little story about fluorophores and reporter genes and how to use them for non-viral approaches, but in principle they're also applicable for viral gene delivery approaches. How can you track them? Where is your material? Where is your transgene expression? So when it comes to light and to wavelengths, the near-infrared area of the spectrum is a quite useful one for such imaging aspects, because you will have a very low autofluorescence, and I'm talking here of this so-called biological window, here around 750, 850 nanometers. You have low autofluorescence, you have a deep tissue penetration. So this is just this example. If you take a green laser pointer, you don't see anything. If you take a red one, you will see. Yeah? The light goes through your thumb. You can make, they are quite dynamic measurements, but disadvantage is definitely that these, especially these near-infrared excitable fluorophores, are quite large. They have a kilodalton or something. So for nucleic acids, it's okay. For small compounds, it will influence your experiment. This is one example we did some years ago with quantum dots. By the way, please also visit the poster session, poster number 375 by my colleague Haider Sami. He will give you more insights into these quantum dots and quantuplexes. So this is a mouse received quantum dots. And you see immediately after injection, this stuff is already sticking in the liver. Yeah? What we did next is we used these quantum dots and used them quite like raisins in a muffin. So we built them in the gene delivery particles and also injected them IV. And what we saw is a very rapid redistribution of the material. First the stuff sticks in the lung, and then it's redistributed into the liver area. So here, with near-infrared imaging, you can measure relatively fast events, yeah, that take place. But as we are all not flat, but three-dimensional, how can you bring this analysis into the kind of three-dimensional world? And there are two principles like the fluorescence imaging tomography, FLIT, and the diffuse light imaging tomography, DLIT. So how does it work? In the case of FLIT, you have in principle a light-tight box camera on top, also with some emission filters. Underneath the mouse you have an excitation filter, and here a light source that really moves from place to place. And then you collect the fluorescence light which is transmitted through the animal. Yeah? But how can you do a three-dimensional image? This happens in a way that you have this mouse on a kind of grid plate, and you take a picture every time the light source moves to another position. With the help of a CT or of a laser, you construct a virtual surface of the mouse, and then you can calculate how deep is your signal. We applied this to track the dynamics of siRNA biodistribution. For this, we applied it by microspraying intratracheally into mice, and the dye, called Alexa Fluor 750, is covalently attached to the siRNA, and you see already with 2D imaging there is something happening. So this is the lung area, this is, we are not sure what it is it a kidney? And when we then analyzed it with this three-dimensional method and combined it with CT, so computed tomography and contrast agent, we could clearly show that already after 6 hours you have a transfer from lung to the kidneys. Yeah? And then we also analyzed the urine of the mice, and you could find the siRNA in the urine. You can also quantify this. You can make such kind of three-dimensional regions of interest and make a calibration curve, and then you can tell, okay, how much of your material stays, for example, like here in the lung area, and how much is moving to other areas of the body. So it's a relatively convenient method. The other thing is bioluminescence imaging. You all know it. You can track either your transgene expression, you can also track tumor growth when you label tumor cells first with luciferase, but again, also here, we are not two-dimensional. Also this has a three-dimensional component. And this is due to a biophysical effect. So you have here, this is the emission spectrum of the firefly luciferase. So it peaks around 500, let's say 60 nanometers, but ranges from a bit above 500 up to 700 nanometers, just with different intensity. At the same time, you have here absorption due to hemoglobin and other body fluids. So if you have your cells just on a plate, you will detect the full spectrum. If you have it subcutaneously, you will lose the green part of the blue-green part of the spectrum. If your signal is even deeper inside the tissue, you will lose the green, yellow, orange part of the spectrum, and it's redshifted. And then you do apply a relatively simple trick. You just measure the emission with relatively narrow bandpass filters, and from this information, together with the surface that you've generated from this animal, you can calculate this three-dimensional distribution of your signal inside the animal, and also here together with morphological imaging with CT. Another last point is that in principle you can also use far-red near-infrared reporter genes, which can then be tracked with this FLIT method. And in the Vakusha lab there have been great developments a few years ago, and they have developed fluorescent proteins that emit at 720 nanometers, which is already quite useful for in vivo imaging. So what we did is we generated a lentivirus, stably marked these tumors, and applied this in a metastatic breast cancer model, syngeneic in bulb C mice. And what you see here is, this is the fluorescence signal in the lung of the tumor lesions, yeah, and here in the background the CT, and you can we also confirmed this by histology that you have such nodules inside the lung. By the way, this is also published now in this Human Gene Therapy special issue. We next then wanted to find out, okay, is it possible to correlate on the one hand the fluorescence signal from the tumor cells and use a reporter gene, in this case luciferase. We just sprayed these polyplexes in, and yes, it was possible. The signal was a bit low, so we did two-dimensional analysis, but like here, you see that whenever you have a signal from the tumor, you also see that you get transfection here with bioluminescence. So taken together, and to wrap it up, such synthetic delivery systems, when you roughly divide it in lipidics, cationic lipids and polycations, have some similar but also different mechanisms of transfections. It's also important that you keep in mind this dynamic interplay between these two components and the cellular environment and the cell itself. You can target them when you attach cell-binding ligands, and such macromolecules can also be tracked in vivo when you attach fluorophores, or you can measure transgene expression nowadays also for bioluminescence and fluorescence also in 3D in vivo. And I would like, of course, my group, they're all really hardworking, like Haidar Sami, he's a group leader for nanomaterials, he's presenting this poster 375 here. Then PhD students and, of course, all our collaborators and our funding agencies. And I would like to thank you for your attention.
Thank you very much, Manfred, for taking us to the journey to the area of non-viral vectors. Are there any questions? Maybe I start. So I'm a viral vector person. When we see this here, the synthetic vectors, where you can put all the modifications onto the vectors to optimize them. So how far away are the synthetic vectors from becoming a viral vector, let's say it like that, with regard to the interaction to the cell?
There's one thing I always wanted to emphasize. Make them as complex as necessary, but as simple as possible. Yes, there are some of them already in clinical trials. Some are more efficient, some are less, but still the efficiency is still a definitely limiting factor. And my guess, really my rough guess, is for localized applications, yes, it will work really good. I'm not entirely optimistic that systemic application will always work, especially when you need a very efficient expression. But what you can do is, it depends on the choice of your therapeutic gene. If you have something that is, for example, excreted and has a bystander effect, then you have a fair chance also to be successful with a non-viral vector in the in vivo application, and then maybe also in further towards clinical application.